The process of mitosis and meiosis. Mitosis is a type of cell division in which one cell, the mother, divides to produce two new cells, the daughter, that are genetically identical to self. Stages of mitosis. We have the prophase. Prophase is the first phase of mitosis, the process that separates the duplicated genetic material carried in the nucleus of parent cell into two identical daughter cells. During prophase, the complex DNA and proteins contained in the nucleus known as chromatin condenses. Prometaphase is the second phase of mitosis, the process that separates the duplicated genetic material carried in the nucleus of parent cell into two identical daughter cells. During prometaphase, the physical barriers that encloses the nucleus called nuclear envelope breakdowns. Metaphase is the third phase of mitosis, the process that separates the duplicated genetic material carried in the nucleus of parent cell into two identical daughter cells. During metaphase, the cell's chromosomes align themselves in the middle of the cell through type of tug war. The chromosomes which have been duplicated and remain joined at the central point called the centromere are called sister chromatids. Anaphase is the fourth phase of mitosis, the process that separates the duplicated genetic carried in the nucleus of parent cell into identical daughter cells. Before anaphase begins, the replicated chromosomes, called sister chromatids, are aligned at the long equator of the cell on the equatorial plane. The sister chromatids are pairs of identical copies of DNA joined at the centrosomes. Telophase or cytokinesis is the fifth and final phase of mitosis, the process that separates the duplicated genetic material carried in the nucleus of parent cell into two identical daughter cells. Telophase begins once replicated pair chromosomes have been separated and pulled to opposite sides, poles of the cells. Meiosis is a process where a single cell divides twice to produce four cells containing the original amount of genetic information. Stages of meiosis. Prophase 1 is a process that involves 5 different stages during genetic material in the form of alleles crosses over and recombines to form non-identical haploid chromatids. We have the spindle formation and synapse homologous pair of chromosomes. Metaphase 1 encompasses the alignment of pair chromosomes along the center of cell, ensuring that two complete copies of chromosomes are present in the resulting two daughter cells of meiosis. Anaphase. The stage is characterized by the movement of chromosomes to the both poles of meiotic cell via microtubule network known as the spindle apparatus. Telophase is that phase when the chromosomes have finished moving to opposite ends of the cell. Prophase 2 prepares the cell for secondary meiotic division, where two haploid cells eventually form four haploid cells, each containing half of the genetic information previously contained in the original replicated diploid cell. Metaphase 2. It follows prophase 2, which primarily highlights the condensation of the chromosomes and the movement of centrosomes to polar regions of the cell. Anaphase 2. The sister chromatids are pulled apart by the kinetochore microtubules and move forward to the opposite poles. Non-kinetochore microtubules elongate the cell. Telophase 2. the chromosomes reach opposite poles. Cytokinesis occurs. The two cells produced by meiosis divided to form four haploid daughter cells and nucleus envelopes. Thank you.